Hello. Thank you for taking the time to be with us here today. So yeah, my name is Guillermo Friis, and I'm going to be presenting the result of our study that here we have titled Evaluating Genetic Diversity and Tree Condition in Planted and Naturally Colonized Young Woodlands. As I'm sure that you are all aware, there is a global initiative underway to increase forest cover, to protect biodiversity, and to help mitigate climate change. Tree planting has emerged as a popular strategy for supporting or accelerating the natural regeneration process. And in this study, we aim to understand if the natural variability that we find in natural populations is adequately and sufficiently captured in planted woodlands, in naturally colonized forests against planted woodlands in this particular study. And in doing so, we hope that we can draw conclusions about their resilience and their adaptability to current and future climatic scenarios. Our goals are threefold, specifically speaking. First, we want to assess to what extent the climatic niches of seed sources, the forest stands that we use for seed collection for planting, sufficiently represent the range of climatic conditions that we see in the wild distribution of the species. We ran this test for a set of 46 UK native species. Then, we focus on two of them, pedunculate oak and silver birch, and we explore patterns of genetic variability across age cohorts, comparing planted populations and naturally colonized young woodlands. There are three specific aspects that we test with this data. We first test for genetic bottlenecks, potential genetic bottlenecks that may result from afforestation protocols and can result in genetic loss. We also test for elevated inbreeding in naturally colonized populations that can result from high habitat fragmentation, as well as for higher co-ancestry in planted populations that may be evidence of a limited use of seed sources. And finally, within this block, we also test for genetic homogenization, or in other words, we basically test if planted populations are more similar to each other than natural ones. And finally, and this is the part of the presentation that Nadia was going to present, we test for differences in health indicators and tree condition. I'll go through her slides to the best of my skills, but we will probably skip a little bit of that part. So, what are we looking to compare? Current afforestation strategies in the UK to meet conservation goals can be broadly classified into two main types. Afforestation pipelines based on natural colonization, where tree communities establish through natural dispersal in former arable or farming lands, and afforestation pipelines based on active planting of commercially produced trees. Each strategy involves different processes that may affect the levels of genetic diversity in the resulting populations. So, for example, in wild populations in natural colonized sites, parental trees may arguably harbor a broader genetic pool and be better locally adapted than commercially or, let's say, like geographically distant seed sources. However, natural processes such as habitat fragmentation, predation, pests and diseases, even climate change can reduce the diversity in these populations through differential mortality. Conversely, planting protocols that integrate multiple seed provenances and aim to maximize survival, germination and survival through highly favorable conditions can, in theory, result in more diverse populations than natural ones. And it is possible that these populations are better able to keep pace through natural selection with emerging stressors. But seed collection, nursery, germination and cultivation, planting, all of these stages are parts of the process that can introduce biases, genetic bottlenecks, and reduce the diversity of these populations. So for the first goal that I mentioned in the previous slide, testing for climatic niche differences between commercially produced trees and wild populations, we focus on these two groups, wild populations and registered seed sources. In this analysis, where we use only environmental data before we move on to the genetic analysis, the idea behind this analysis is that if we have a number of populations that are adapted to different environments, but we rely only on a few of them, we may be losing, for planting, for seed collection and planting, we may be losing some of this adaptive variability, right? By using high precision seed source location data, which is commercially sensitive and has remained private through the study and the publication, we can build highly accurate climatic profiles. And using this data, we can compare the climatic ranges of seed sources and natural populations to assess how well seed sources represent the full range of climates experienced by the species. Results for this analysis can be visually explored. So what you see here is a spatial representation of the climatic range of seed sources in green and of the wild distribution of the species in orange for two examples, pedunculate oak or silver birch here. So what is important to notice here, this is based on principal component analysis of environmental data, but what is important to notice is that the climatic range of the wild distribution of the species consistently represent or covers a broader range of climatic conditions. And this was the case for 31 out of 46 species that we tested. And they were statistically significant, these differences. So this result suggests that seed collection in the UK for planting may be often overlooking the full environmental variability that we find in the wild, and this may reduce the adaptive potential and resilience of the planted populations. So moving on to the genetic analysis, in this study, as I already mentioned, we focused on two species, pedunculate oak and silver birch, and we studied 3 equivalent age cohorts in the 2 different strategies for afforestation that we are studying. We study wild populations that are parental trees, putative parental trees in natural colonization sites against seed sources, and then we also study seedlings and saplings in natural colonization sites against nursery seedlings and outplanted saplings. We started by testing for differences in genetic diversity using a number of metrics. I'm showing here 2, observed heterozygosity and nucleotide diversity at the bottom of the figure, which is represented by pi. So let me use this figure just to explain a little bit about the design of the sampling of this study. For natural colonization pipelines, we sampled 4 different populations where natural colonization was ongoing, represented by different colors. You can see the bar plots there. And we sampled these 3 age cohorts that we're interested in, shown above the figure. In the case of planting pipelines, we sample a variable number of seed sources, 1 orchard in the case of birch, nursery trees of different origins from 2 different nurseries, and then 3 outplanted populations per species. So what is important to notice here, the pattern that was relevant from our analysis, is that very minor genetic differences in terms of diversity were found in between groups. Even when we compare natural colonization pipelines and planting pipelines, the levels of genetic diversity were very similar. And this is important because we can infer then that planted populations do not show reduced diversity due, for example, to genetic bottlenecks or artificial selection, but they don't show either an increased level of genetic diversity that could derive from seed mixing or from favorable conditions at the early stages of their life. Next, we tested for inbreeding across age cohorts in both wild and commercially produced trees. So inbreeding is basically the potential loss of genetic diversity as a result of reproducing between 2 closely related individuals. And in this analysis, in our analysis, we measure it using a statistical called FRO. And what we found is that seedlings and saplings in natural colonization sites showed higher inbreeding values in both species than any other group, higher than the parental trees in the very same sites, higher than any group in the planted pipelines. And this is relevant because this is an evidence that habitat fragmentation and isolation may actually be resulting in inbreeding in natural colonized sites. And it also emphasizes the potential role for assisted gene flow, for example, in planting programs to maybe alleviate these patterns and the associated risk of losing diversity. Next, we tested whether individuals within planted populations were genetically more similar to each other than in natural populations, and also if planted populations were more similar between them than natural ones. And to do so, we computed levels of co-ancestry. High co-ancestry is not the same thing as inbreeding. Co-ancestry basically measures how closely related an individual is to another individual in a population or across populations. The concept or the key link in these 2 concepts is that a population where high co-ancestry occurs is at risk of producing offspring, subsequent generations that are inbred. And that's the risk that we are trying to assess here. I'm showing the results here for oak in that heat map. It's a rather complex figure, and I'm always afraid that it's not that easy to understand when I present it, so let me explain just a little bit with a schematic version of it. What we have here is a double entry table where populations are grouped, are arranged in columns and rows, so that in the diagonal, each population is compared to itself, and that's where we record within group co-ancestry values, and in the remaining cells we have all the pairwise comparisons in between the populations and groups for natural colonization and planting pipelines. So there's a number of patterns here of interest. The first one that we found relevant was that the levels of co-ancestry within group in nursery trees and planted populations were generally higher than those occurring in wild populations. And this is worrying because, as I already mentioned, this puts these populations at risk of generating inbred offspring in the next generations. The next pattern that was worth commenting for oak is that when we looked at the pairwise levels of co-ancestry in wild populations, these values were consistently and significantly lower than those are represented in the bigger triangle at the top of the figure. They were smaller or lower than in the case of comparisons between planted populations, which are in the smaller triangle at the bottom. Which means that there is some degree of genetic homogenization, which is reducing complexity in the landscape in the UK for the case of oak. It's worth mentioning that we didn't find this pattern, or not to the same extent in the case of in the case of birch, which is presented here, among other things because we found in our data set that more diverse collection of origins for seed sourcing and planting was used in the case of birch, and that seemed to keep basically the differences between populations. Furthermore, the lowest level of within group co-ancestry was found in the only orchard that we have in our data set, and this suggests maybe orchards are a good strategy to deal with this problem, to basically prevent this from happening, this higher co-ancestry in planted populations. This was Nadia's part, in which they basically measure different health indicators such as mildew impact or leaf spot in birch, comparing planted populations with natural ones. I'm going to go very quickly over this because, yeah, I don't even know that sure what happened, but basically they found, for example, that mildew was less likely in planted oaks, while leaf spot, which is a fungal infection, was more likely in birch populations, in planted birch populations. So that's basically it. Let me just wrap up with the main points, the main findings of my study. First, we found that for many UK tree species, commercial seed sourcing doesn't fully capture the climatic range of conditions that the trees experience in the wild, and this may reduce the adaptability of planted populations. Second, we found that genetic diversity levels were generally comparable between planted and naturally colonized sites. However, natural colonizers showed higher inbreeding, which may be the result of habitat fragmentation and isolation, while planted populations were more genetically uniform, reflecting maybe limitations in seed sourcing. And third, which is what I just mentioned from Nadia's slides, overall, both planted and naturally colonized trees were in good conditions with only minor differences, but planted oaks were less likely to have mildew, while planted birches were more likely to show leaf spots as a result of a fungal infection. Thank you.